Okay, thank you all for coming today. We really do appreciate you guys taking a couple hours out of your day and coming to learn about this emerging issue that we have. Hi, Bill. We really are excited because we think today's educational session is a good first step in maybe a regional response or a regional approach to how to address this issue in our county as well as more regionally. And I wanted to give you a heads up on today's format. We've got one speaker, Dr. Swiecki, who's going to speak first, and then we'll have time for a few questions and answers in between the two speakers. And then our second round of speakers is a tag team duo that's speaking from SFPUC, and then we'll have time for a few questions and answers afterwards for all three speakers together. And one other thing I wanted to mention to you, if you haven't signed in, I'd appreciate it before you leave if you could sign in so we can keep track of who actually was here today. And thanks again, all of you, for coming today. So without further ado, I'll introduce Linda.
So our first speaker is Ted Swiecki, Dr. Ted Swiecki. Ted earned his PhD in plant pathology at UC Davis a while ago. I won't say when. It's back when I was there. His doctoral thesis was on Phytophthora, and he has continued to research diseases caused by this genus throughout his career. He is principal of Phytosphere Research in Vacaville, California, a two-person business started in 1987 with his partner Elizabeth Bernhardt, who also has a PhD in plant pathology. So this is a dynamic duo. Luckily for us, Ted and Liz have sidestepped working in the ivory towers of academia and instead have trekked out to the wilds of California to study distressed, charismatic megaflora like oaks, native oaks. Their research funded by the U.S. Forest Service has resulted in two very useful free publications, which I just want to mention quickly, which you can download online, which are ideal for field-oriented staff. One is a reference manual for managing sudden oak death in California. That was a 2013 publication, U.S. Forest Service. And the second is a field guide to insects and pathogens of California oaks, also U.S. Forest Service, 2006. Today, Ted is going to tell us about the modus operandi of several Phytophthora plant killers and why increasing our vigilance and preventing their spread into our open spaces is warranted. Please welcome Dr. Ted Swiecki.
Okay, thank you. So this slide's been up there for a long time, so I don't have to talk about that. That's my title. Okay, so we're going to do a little bit of this myth or fact format. And so some people may come to this meeting with some basal knowledge of Phytophthora, others of you haven't. But a lot of people that know a little something about Phytophthora may think of them as root rots in over-irrigated landscape plants. Unfortunately, that's not the only issue here. As we'll show in the next few slides, that these root-rotting Phytophthora species can be lethal to a wide variety of plants, including in very dry habitats. Janelle likes to show pictures of her favorite endangered species. This is one of my favorite endangered species, Arctostaphylos myrtifolia, the Island manzanita, occurs in the foothills of the Sierra Nevada and the Ione area, if you've ever been there. This is what it should look like. This is what it looks like in a lot of areas where Phytophthora cinnamomi has been introduced. It's a completely lethal disease to both myrtifolia and the other co-occurring species here, the taller species, Viscida. And it is basically destroying these stands at kind of an alarming pace. The plants have really no resistance to them all and pretty much are just being decimated in these areas. They grow on these particular soils that are high in acidity, and actually not a lot of other things do grow on that, so that's why a lot of the areas where you had mortality, nothing ever grows back. Here's a patch where we can see just it's about to die off. We've had more older mortality there, but this intervening patch is about to go. Just to give you some area of the scope, this up in the corner here is the city of Ione, which isn't a very big city, but all these areas here are completely infested, and that's just a fraction of the area that is occupied by this pathogen that is basically destroying this particular plant species. So we started working on that in 2002. A couple of years later, we got a call from Fish and Wildlife, Cal Fish and Wildlife, which would be Fish and Game back in the day. And they mentioned there was another Arctostaphylos pallida in the Oakland Hills, an even rarer species in that respect. They'd seen some issues with. As it turned out, we did find that Cynomolmy was also affecting that species, and along with it, this giant chinkapin, which is growing with it. In that area, a couple of years later, that's above the Huckleberry Botanic Regional Preserve. Problematic in that that preserve is partly there because of that particular species. We also got a call a few years later, another population near Joaquin Miller Park also is dying out with the Cynomolmy we identified in that site. Well, we've been working on sudden oak death at a number of locations in Marin County, including China Camp State Park. And we hadn't really paid that much attention to some of these dying madrones there, partly because they aren't killed by sudden oak death, and we had enough trouble with the sudden oak death. But we actually found out, once we've been doing all this other work, we thought, you know, why are those madrones dying? Well, it turns out they were dying also because of Phytophthora cynomolmy being introduced there. One of the odder things that eventually caught our attention was that these areas where the sudden oak death had killed all the oak overstory, you would think the bays would have been doing great because the competition would be reduced. But as it turns out, the bays were dying, and that was, again, because of cynomolmy, which had been introduced in that area. We saw here, and we've seen this in several locations, we have the oaks dying due to sudden oak death, Phytophthora ramorum, and then we have the other species dying because of Phytophthora cynomolmy, a soil-borne pathogen. So that doesn't leave a lot when you're done, and it leaves a kind of a problematic situation there. It's been spreading out. These madrones here are just dying. They're just turning color right now. In that particular site, we have this whole area on the slope that was infested, but we did some sampling with Liz Fitchner from the Rizzo lab, and we also picked it up higher up on the watershed. And as I'll mention, that's problematic because these soil-borne Phytophthoras move downslope fairly readily. So whenever you get it high in the watershed, it can move downslope a lot more easily than it can move upslope. So everything downslope in the watershed is potentially at risk. We've been working with PUC on the Peninsula watershed on some sudden oak death related projects. They had also pointed out a patch of a mortality of madrone on their watershed, which also turned out to be associated with not only Phytophthora cynomolmy, again, the signature here of both dying madrones, dead and dying madrones, and declining bays. Bays die a little more slowly, so that you usually have the madrones dead first and the bays going out. But we also found, as we'll talk about a little bit later on, some other Phytophthora species in that area, including Phytophthora cambivora. MMWD, Mount Tam watershed, again, place where we're working with sudden oak death, but we had some madrone mortality in this area, where we also confirmed Phytophthora cynomolmy was killing these madrones, including these areas like this. So it's pretty, it can be pretty devastating in areas where it gets established. This is a trail on the Mount Tam watershed, though on Marin open space lands, where there's a mortality center, a giant chinkapin, again associated with Phytophthora cynomolmy. This, the one slide is taken looking down from the trail, the other side is taken looking up from the trail. So that it's evidently brought in on the trail and formed a mortality center that spread out from that introduction site. More recently, we were working on some sod-related fuels issues with Jack London State Park. Did see the signature again of dying madrones and bays. Found that both cynomolmy and cambivora associated with mortality of those species and some others at that site as well. This is an interesting site we were called out to look at in the Miacamas Mountains. It's a private property. But they had these small patches of dieback. But they actually, a lot of them were associated with areas where there had been perk tests done, trenches that had been dug to check percolation associated with various things they were doing there. We did find cambivora associated with the mortality in that site. And what we try to point out there is that when you're moving soil, and this is just a tracked vehicle that actually was parked there, you can see the amount of soil that can be moved on these vehicles from place to place. So if you get an infestation in one area and you've got this much contaminated soil going, it's going somewhere else pretty soon. Another site on the PUC property here, coast live oak and coffeeberry mortality associated with the taxon PG chlamydo. Some of these species don't even have names yet because they haven't gotten around to naming them. A lot of them are fairly recent. But this was kind of problematic. This was also next to a roadside, internal road, drainage area, so the possibility of introduction there along that road certainly possible in that site. And we throw this one in to show you it can also affect conifers. This is Cryptogaea associated with these dying, with a plantation of Douglas fir on the watershed. So the bottom line is, it's not theoretical that these Phytophthora species can cause problems, and they're not limited to one single geographic area. These are some of the places that we've found mortality associated with Cynomolmi and other soil-borne Phytopthora in Northern California, and those largely coincide with places where we've actually looked for it. You have to look around, but if you look, you can actually find it in more places than you would hope to. So some people think, well, aren't they just native, or aren't they everywhere to begin with? And that's really not true. All these destructive Phytophthora species are introduced. None of these are native to California, and probably most of them are not native to North America. And they have been found in nurseries, landscapes, agricultural fields, but these are all exotics that work their way into the wildlands through these various routes, which we'll be talking about. And one way to also think about, well, how do we know it's not, you found it here with the dead plants, how do we know it's not over there where the live plants are? Well, in the Ione Manzanita sites, we had set up some permanent transects where we were actually looking at the distribution of inoculum. As you can see, most of the inoculum we found that was viable was right at the 0 meter line, right at the interface between live and dead material. As we went even 2.5 or 5 meters into the live stuff, we didn't recover the pathogen. We get into the dead stuff, it's been dead for a while. We have a little lower recovery because there is some loss in the number of propagules over time, although not fast enough to do us a lot of good, as I'll mention. So you can see that line is pretty much where the pathogen is. So it's pretty good in this case with this very sensitive plants. You have a pretty good bioindicator. This is that same transect 10 years later. You can see all these areas which weren't infested before have subsequently become infested. This is an upslope direction, so running water isn't involved here. This is root to root spread. You can see it's well beyond the 5 meter point at that point. So you can get an idea of how these things spread out over time. And so one of the questions is how they're getting there. And this is a site in Sonoma County where you can see these dying bays here. It's next to a home site. We did sampling around this area. We did find Cynomolmi associated with the dying bays and madrones in this area. Even farther down the driveway, away from this mortality zone, we didn't pick it up. So clearly some introduction there, probably associated with a lot of the landscape plant material here next to the home site. This is some sampling we did on the PUC Crystal Springs watershed. I want to point out a couple of things here. These clear or round white circles are negative samples, and you can see we're sampling in a number of areas here looking for symptomatic material. And it's not every place you see dying plants, but there are definitely places and clusters, as you'll see in here, where we do pick up a lot of Phytophthora species. In particular, this area near the Philoli estate was very hot for actually a variety of Phytophthora species, including Phytophthora Cynomolmi and Cactorum and Cambivora, as well as some other ones. Well, what's special about this spot is here's the Philoli Gardens and estate. Here's a caretaker's house. The road goes right between these, connects these horticultural operations. So, and it's well known to the people here that they've got Phytophthora issues in their landscaping stuff. So it clearly spread from that landscaped area into the wildland there. The issue there then is to prevent further spread along the road there, because we have an unsurfaced road in that area. There's mowing that has to happen on the margins of that. So if that happens during wet conditions, it can spread that contaminated soil along those roadways. Certain places, once we've found issues there, have responded. This is China Camp State Park. They actually closed one of these trails due to this infestation. They actually obliterated the trail entirely. This is from the Ione area where BLM has a site where we did find infestation as well, and they've closed that area to public use again to try to prevent further spread by vehicles and people getting there, especially when the soils are wet. So, so this is the question. So are these things that kind of a flash in the pan? Are we going to, well, the plants will get over it eventually? Well, that's really not the case. Over the short term, as we would say, it's not bloody likely you're going to get any kind of resistance to these things, because what we have are naive populations of plants that did not evolve with these pathogens at all. A lot of these are from Asia and have been separated for millions of years from their progenitors probably. So that's not going to happen. Even in the long term, there's not any guarantees that these native plants are going to have anything that will get them over the risk. We do see, this is an area where, with this Ione manzanita again, this is a site where we have some regeneration occurring where there had been mortality. And this is a little bit later that same year. That plant is dead now. You can have temporary escapes if these plants don't hit inoculum, but as long as the inoculum's there, they basically intercept it again and are killed. So that's what we see here. Here's some old dead skeletons of Ione manzanita. All this little brown stuff here, those are all regeneration that's been killed. This one isn't quite dead yet. And so you see in a time lapse like this, we look up this old road, which might have been the original source of introduction into this area. You see expansion of mortality. You don't see anything else coming back in those sites because it just doesn't, it can't, there's too much inoculum there to keep it from the plants from dying. And even if we look at something like sudden oak death, where there is research into whether tan oak shown here is whether there might be some resistance in that population. Maybe there is there's some evidence that there could be resistance in that population. And maybe over time that can be developed. But in the short term, you've got impacts like this, and the short term being next 50 years plus probably. You're not going to restore this forest anytime less than that, and probably not with tan oak because there's nothing to be had there to grow in that situation at this point. So what is this thing called Phytophthora? We'll hear people say fungus, or in the case of the chronicle, you'll see mold sometimes thrown out there. What these are called is a group of mycetes known as water molds. They're really not related to fungi. They're actually more closely related to the algae. They find out in this group here of the golden brown algae. Fungi are often this branch here somewhere. So they really only were considered fungi mainly because the growth form is similar. They have a body, if you will, that consists of fine filaments or mycelium, which we see growing on this plate here. Their cell walls are out of glucans and cellulose as opposed to chitin, which you find in true fungi. And phytophthoras are mostly plant pathogens, primarily dicots. There are some monocots that are also affected. And you're aware of some of these. Phytophthora morum, shown here on tan oak, was only described in the well, actually, it was found. It was actually seen in the 1990s in nurseries in Europe. It was not described till 2000 as the cause of sudden oak death. The species name was only applied after that. But at this point, that particular pathogen has at least 90 known hosts in these good number of genera and families. Phytophthora cinnamomi, which we've been talking about, has over 3,000 known hosts, and it could be a lot more than that. And you can see that's covering over 91 plant families. So it's much more of a generalist. And even Phytophthora cactorum, which we've seen in a number of sites here shown on a replanting material or planting, over 400 hosts, and again, around 90 families plus. And all these numbers are kind of subject to revision because we haven't seen them against all their possible hosts. So we add, every time we look at a native plant, we usually end up adding a host on there. Phytophthora, the name itself from the Latin, or actually from the Greek, plant and destroyer. And it was named after this particular organism, Phytophthora infestans, which was the cause of the great potato famine in Ireland. It was a great social disruptor in the mid-1800s. Also led to a lot of settlement in the U.S. This monument's in Boston. But P. infestans, as it turns out, is actually what we would consider a rather narrow range, kind of relative, wholly host-specific. It only has about 130 hosts in 12 families, and about half of those are different species of Solanum, where the genus that the potato is in. In terms of the number of Phytophthora species out there, there's a goodly number. We keep track of them in databases like this that people keep their sequences on. Clive Brazier, world authority on this, estimated back in 2007 there might actually be around 200 to 600 species, and this is over time, the number of described species, and you can see it's taken this kind of jump up. If we extend it with these things in the current database, we see we are pretty much following his trajectory. So there's a lot of species out there that haven't come out of the woodwork yet, and that's one of the things we're concerned about. Again, the form is this mycelium, each strand of which is referred to as a hypha. The morphology or shape of that differs a little bit between species, and sometimes you can get an idea which species they are based off of that, but not always. They produce several different spore forms. This is for asexual production, sporangia. They can either direct germinate like this to produce a germ tube, or they will release these swimming zoospores. This is my cheap animation, but that's what you see is these things swimming out of these jobbers. This is what this electron micrograph of a single zoospore. They have two long flagella, which they use to swim in the water. And if our video works here, oh, death. Oh, that's interesting. That was there earlier, so that video is not going to go. At any rate, I had a video here. Let's go back to slideshow. All right, was there before the presentation. We'll have to look at it some other time. Anyway, take my word for it. It's like the cheap animation. They swim out of these things. But what happens is that they're attracted chemotactically to host tissue. So all these little round bodies here are zoospore cysts. The zoospores swim, they form a little ball, they germinate, send in a germ tube, which penetrates into the host, as we see in this cross-section, and then proceeds to colonize that tissue and kill it. We also have these oospores formed, or chlamydospores, sorry. This is a type of survival structure we see in some species. Oospores was my next slide. These are both a survival and a sexual reproduction spore, and they can be kind of long-lived in the soil. So to show a quick presentation of what the life cycle looks like, there's a sort of a fast phase of the life cycle that works under wet conditions. Essentially, under wet conditions, the sporangia form, release these swimming zoospores, insist on roots, and within 24 to 48 hours, you can have new sporangia form to release more zoospores. And each of these sporangia can have 20 to 30 or more zoospores in it. And there's a lot of sporangia. If you think about that, you can see how explosive that can be over time. Also in these roots, we have these resistant structures are formed, and those serve for both survival and dispersal under suboptimal conditions. So when it's drier, they survive. They can, when it gets wet, reinfect the roots or form sporangia that will then release zoospores. there's several ways to detect Phytophthora in infected plants and soil. One of the most common ways is to bait with various things, and it turns out green pears are a pretty good bait. This is an example of a couple of soil samples being baited. Here they are in a tray, just kind of so you can see it a little bit better. Water is added to the soil root sample. You need to have a lot of roots in there. Certain leaves can also be used as baits. In this particular sample, we are now seeing this sort of bathtub ring, as we refer to it, around the water line is pretty good evidence that zoospores have attacked all along that, and some of the necrosis showing up on these bait leaves, which then you have to isolate from to get it. You can get sort of semi-quantitative ideas of how much inoculum is in the soil from baiting, from very heavy levels of inoculum where the pear is just blasted down to the couple of oddball spots you have on that there. So it gives you an idea of, is it highly infected or do we have the light infection levels perhaps? Okay, so we've already talked about this whole business about functioning in dry situations. In the nursery, it's often a sort of a shorthand say that people will say, well, you really get Phytophthora from overwatering. And the point is that overwatering does not generate Phytophthora, right? It just provides conditions that it can thrive under. So just as an example, this was a pathogenicity test on our own manzanita plants. All these plants were flooded twice for 24 hours at a time, just 12 days apart. You happen to see one batch are dead, the other are not. Well, you could consider all these were really overwatered, but if we have zoospores in them, they're dead. If they don't have zoospores in them and they're growing in clean soil, that event is meaningless to them. And so then I have to kind of introduce this whole concept of the plant disease triangle, because to understand diseases and how they work in the field, you have to kind of get that. And really, the classic plant disease triangle indicates that for disease to occur, you have to have a susceptible host, a virulent pathogen, and a favorable environment. And it's sort of like if all those things are there, you can get disease, and if one of those legs is broken, it won't happen. As it turns out, you could make it a little more complex than that, because really there are abiotic, weather, temperature, moisture areas, and biotic aspects of the environment. There are antagonists and other things that can make plants less susceptible. So all these things interact, and there's kind of a sweet spot where if they're all in the right balance, disease is more likely to occur. And furthermore, it has to occur for those conditions have to prevail for some period of time for infections to occur, for instance. So we really can make this into sort of the plant disease pyramid, that these interactions all control whether we get infection and whether it develops into a severe disease. So if we look at the kind of conditions that are needed under abiotic environment, we want to have moisture and humidity, well-aerated soils, periodic saturation, moderate temperatures. Those are all very favorable for Phytophthora. Those are also conditions you find in nurseries. Under the host, high host density, high root density, predisposing stresses are also very favorable. Those are also things that are very common in nursery situations. If we think about the root density of a nursery-grown plant, it's far beyond what you would see anything grown in the soil. And in terms of the biotic environment, low density and diversity of other antagonists and microbes, and we find that in nurseries as well, because we're starting with the soil as media, it's less complex. And the time aspect, if we let it go into bigger and bigger stock, like these bigger oaks, we've got plenty of time there. So the bottom line is this: the plant nurseries provide almost optimal conditions for Phytophthora diseases, and that's one of the issues here, is that because it's so optimal, you don't have to have a lot of Phytophthora introduced into the nursery for it to blow up into a big-time issue. So perhaps you can just inspect your plants and say, okay, well, these are infected and those are not. Well, we wish it was that easy, but it doesn't always work out that you can tell the difference. Quite often, disease roots look very diseased. This is a one of these ion manzanitas that was inoculated. You can see we cut away the stem, how necrotic it is. Here's these roots. They look not very good, right? Well, this is a non-infested ion manzanita. You can see it's got a healthy stem. But these roots don't look that much different. You can see there's density different, but they're still about the same color, and they still look kind of pathetic. So in some respects, unless you really know the plant and how it's growing, you can't always be sure whether you're dealing with an infected plant or not. So here's a California bay grown in a pot there. We see some darker roots. We see some lighter roots. Is this infected or is this non-infected? Well, if we compare it to this one, we might have a different thought about it. As it turns out, this was an inoculated. This is not inoculated. That was inoculated with zoospores of P. cinnamomi. But even in an inoculated plant like that, you can see there are some healthy-looking roots. There's some totally rotted roots. So it's not totally uniform. And that's one of the issues that makes it very difficult to inspect in nursery and say, okay, these are healthy, good, these are bad, throw them out. It's not quite that easy. And there has been kind of the underlying assumption, I think, for a long time that Phytophthora root rots were not a problem in native plant nurseries. As it turns out, to our chagrin, this is not the case. And as a lot of work done, especially in the last year, has really shown how many Phytophthora species are found. This is actually work going back several years, done by Elizabeth Fitchner with us when we were doing the pathogenicity to add madrone essentially to the cinnamomi host list. It wasn't on there. Well, she had to get some plant material to continue these tests. Well, one thing she found was that a lot of the plant material she got, she couldn't use because it's already infested with a number of species, including P. cinnamomi, the one we were trying to test on it. She actually had 8 Phytophthora species isolated from a very small number of plants that she bought as healthy plants from native plant nurseries. So that didn't bode very well. And so when we were working with PUC and they told us a little bit about their restoration project, which you'll hear much more about today, we suggested that they might want to put some practices in place to try to avoid making sure that they don't end up with infested stock in these large plantings. And they'll talk about how those things worked. But the net result was it didn't work all the way. And in 2014, we got a call that some toyons looked a little iffy, and some people thought, well, maybe it was frost or whatever else. As it turned out, there were 2 species of Phytophthora in these newly planted toyons, including Phytophthora tentaculata. Basically a quarantine species, number 5 on the list of Phytophthora species that were in an analysis put down as Phytophthora species that were threats if they were introduced into the U.S. And this first showed up a few years earlier in a California native plant nursery. This was only a second detection of it, again in California and associated with native plant nursery. Here's a toyon from that same nursery that wasn't planted, where we had Cactorum. One thing I want you to notice is these roots, I will tell you, are not healthy, and there are also not very many of them. But you will see that there's actually still some green leaves on this, and this is only just recently turned brown. And that's one of the issues with California native plants. A lot of them are so drought tolerant that when the root systems are totally rotted out from under them, the tops still look good. So you take delivery of a bunch of plants, they look pretty good. They start dying in the field, well, maybe they didn't get enough water, whatever else. Well, the other possibility, and it turned out more common than not, these things are actually infested with Phytophthora species, which are killing them. So we went and looked at some of the other nursery material that was scoped for the site. This particular nursery, we found Phytophthora quercitorum, which is, I think, been only rarely reported in California. I think there's only 1 or 2 other reports. As the name implies, it goes to oaks, and it was found on a coast live oak, not necessarily something we want to really introduce into our oak woodlands. At this nursery, we're looking at the plants they had left to plant, and there really wasn't a lot of things that we would consider Phytophthora hosts. Carex aren't really known to be Phytophthora hosts as monocots, but they had some divisions here that didn't really take, so we sampled them. Oops. And as it turned out, we picked up Phytophthora plurivera in that material. So that would be a new host report. We don't know whether it's just infestation there or was it really associated with whether it's just the soil contamination or whether in fact the plant was diseased as well. But not a place we even expected to find this pathogen. So maybe we plant the stuff in the field that won't survive. We could hope for that, right? Well, no such luck as it turns out, because where we've seen this material planted out, we can detect it, and in many cases, a year or more after planting. Here's a planting that was done. This is on PUC land. Here's some twines looking pretty funky. We picked out Phytophthora cactorum out of these. They were still in these little grow bags here where the roots had really not gotten out, and the reason they hadn't gotten out was because they were being rotted off by the Phytophthora. Here's some mimulus that have been in the ground for more than a year, where we picked up Phytophthora megasperma and cryptogaea at this particular site. This site was really problematic because we know the fact that these Phytophthoras really like wet conditions in terms of spread, so that's kind of not a good situation to see them in. And we did pick up megasperma and inundata, which was originally described in the UK affecting willows. And so that was a species that we were very concerned about also getting into a wetland site. At the same site, we actually were able to bait this water here and come up with cryptogaea in it. This site is downslope from, or downstream from where they had their holding area for the nursery plants. So potentially introduced into a wider area there. At this restoration site where there were thousands, and Ellen and Greg will talk about this, thousands of plantings, one of the issues was the mimulus, which is kind of a favored host of Phytophthora tentaculata. And there were some plants that were in the ground for 2 years, or they'd oversummered, overwintered in the ground. They still looked good, and then they started dying. And we picked up tentaculata associated with them, so we know that that species is capable of surviving at least over a full growing season in the field. Just some other ones we picked up recently. This Anaphyllus, this is a pearly everlasting. It's like, who would know? Anyway, P. cryptogaea on the roots of this, as well as on some mimulus at the same site. This is a planting in a basically near a parking lot that was created on the peninsula. And this actually just in Vacaville annoyed the heck out of me. This planting done by the RCD, where they put some material, some mimulus in, which really don't even belong on that site. We sampled 2 plants, and we got 3 species of Phytophthora, Nicotiana, Megasperma, and Pini, of all things. We didn't even bother to sample these other ones because it was just kind of a demo, but it begs the question of how many other species we could pick up if we did more sampling there. I mean, that's big time bad in terms of infestation levels. So this is a little breakdown just showing some of the species that have been detected on some plant species. The main thing I want to point out is that all these red species are ones that are rare, or maybe in some cases they may have one other detection in California before we found them in these various things. We found these things in field situations and in nursery situations. These blue colored plants are all monocots. They aren't even normally considered hosts, and we're still picking Phytophthoras up on them. So not a good situation, implying that there's a lot of infestation in these different nurseries. We could score them by their pseudonyms here. And we can see that some of these species, the more samples we take, the more species we get, basically. So there's a lot of them out there. And a lot of these species of, rare species of concern are showing up in these nurseries. So one thing people say, well, why don't we just spray these things with fungicides? Won't that take care of them? Well, that is not the case, because fungicides, despite the name, in fact, fungicide sounds like herbicide and insecticide, which are things that kill weeds and insects. Fungicides really only should be called fungistats because they really only suppress fungi. They hold them in check, but they don't usually kill them, especially if they're associated with host tissue. So what happens is that they can become unsuppressed when you stop applying the fungicide, and they're back into town. Also, there's issues about sensitivity and dose. This is Phytophthora cinnamomi. We did a test on an area where they've been applying a lot of this particular fungicide, metalaxyl, and we have tested different rates of the active ingredient here on plates, and you can see how it's suppressed at different levels. The ED50, or the 50% growth rate reduction rate, is down here between about 0.27 micrograms per mil, somewhere between these two numbers here. But if you actually look at what kind of residue tolerances there are for these, like in crop plants, you see that this is the types of residues you normally find in treated plants, okay? And you see they only go up to about here, where we have growth out to a lot higher. So the bottom line is you would have to apply a ton of this material to suppress it even down to these higher levels, and that'd be well beyond what use levels would be. Also, in this particular material, if you apply it repeatedly, you can develop resistance in the pathogen. And so basically, fungicides aren't a cure. One thing they do is they kind of interfere with detection. So we had these samples that we looked at, and we thought, these are dead ringers. We should pick out Phytophthora and these rotten things for sure. But this particular nursery does use one of these fungicides, and that basically suppresses it enough that you won't be able to, if it's recently treated, you're not going to be able to pick it up in your sampling. So effectively, you can game the system, if you will, by dosing your material with fungicides. The plants aren't clean, but you might not detect it. So that's a problem in terms of trying to figure out clean ways to handle this. Ideally, you want to start with everything clean potting soil, clean pots, etc. So we think we can test our potting material or plants, and they will know whether they're free. And the problem is that this isn't quite so simple. The main point is that all of these tests can generate false negatives. Any single test you do can give you a false negative. And there's a number of reasons for that. Some of it's just the sensitivity of the test, that you can't detect below a certain threshold. Other things have to do with when the material gets infested. So if the potting media is clean at one point, and then you test it then, and then other things that happen in the meantime, that test may not mean anything in the future if you have introduction later on. As I mentioned, interference from things like fungicides can occur. And if your plants are either just recently infected or they've been dead for a long time, those are both situations where it's very hard to detect the pathogen. And part of it becomes the sampling issue. So again, here's an unnamed small native plant nursery, which even if this material was clean to start with, there's no way they can avoid getting it contaminated just because people are walking in off of this, stepping in the place where you're shoveling your soil from. So inoculum could easily be introduced into that material. And the question is, well, why can't you just detect it in soil by running a test? It's the needle in the haystack issue. Basically, if you're going to find a needle in the haystack, you pretty much have to take that whole haystack out, take it apart, put it into little pieces, and then do something to detect each of those pieces where you might be able to detect that one needle. Well, what would that look like? It would look like this, which also looks like people growing container plants. So in fact, when you're growing container plants in a nursery, you're essentially doing what we might consider is a good large-scale bioassay for Phytophthora. So in the end, you're never going to be able to test your soil clean, so you just have to make sure your soil is clean to begin with. Because the bottom line is you will find it, but you'll find it by means of having infested plants, which you may not detect until it's then spread to a lot of other material. Also, just to sort of show the detection issues here, this was the site where we had the manzanitas with the Cambivora. We saw a lot of symptomatic plants in some of these areas. Some of these, it may have been Cambivora, some cases maybe not. But we actually only had 2 positives there. And these 2 positives, one was negative on the first sample and one was negative on the second sample. We actually sampled these 2 spots twice. And this one was negative on the first try, positive on the second one. That was positive on the first try, negative on the second one. So you're going to have low levels of inoculum you can have this kind of just random error that you can just miss things sometimes. So a single test is less informative than multiple tests, but the fact is a negative result gives you not so much information in many cases. A positive one always has information because they don't come from anything. Okay, back to the nursery situation, some of these issues. Talking about getting plants off the ground is one of the BMPs, the best management practices. And if people have plants off the ground, they think they can't be infected with Phytophthora. Well, you have to get, it helps, but you also have to have some other things going on, including enough difference from the ground. So in this situation here, we can see the imprint of things that were on the ground here at one point. So if you simply say, okay, we're getting all our stuff off the ground, you take it off the ground and put it onto some low pallets, well, if it's already been infested when it's on the ground, it's not going to do any good because you're just moving your infestation up to the pallets. But also, if we just look at the dynamics, physics of splash dispersal, you can have, if a water drop hits, from a raindrop hits the ground, and you look at the splash, you can get about a meter to 3 meters horizontal spread, about a ½ meter good vertical spread. So if you're only 10 centimeters off the ground, a good rainstorm comes through here, all the kinds of stuff that's in this aisle is going to be splashing onto these edge pods and stuff. Okay, so that's an issue, and also we've talked about once you have contamination in the nursery, it can spread. Here's a situation that was in a nursery. Here's the guy throwing out the diseased plants next to the clean soil pile. So there's some kind of problems that there have been. There's these issues about whether it's a quarantine level species or not, and maybe we're only concerned about the quarantined ones. Well, as it turns out, quarantine does not equal impact. And some of the most impactful species like Cinnamomum are not quarantine species, but they're very, very problematic. So part of the issue here is this. If we're doing ecological restoration, this is a planting of oaks all done from acorns, so no risk of pathogen entry there. If we're introducing exotic pathogens, we're kind of not doing restoration anymore, okay? Because restoration means you're putting something back how it used to be or in a better condition. Introducing an exotic pathogen doesn't achieve that. So that pretty much sends that whole bit down the tube because you also have the impact that not only is your material putting out there going to have problems, but you may affect the existing vegetation at the site. So kind of to summarize here real quickly, some of the reasons we should be concerned. First off, we know that these Phytophthora species are already affecting a wide variety of plant communities in a lot of different climate types, soil types, so it's not a rare and unusual thing to have them go. We've got both rare and common native species that can be affected. Once we get an infested area, we can start moving it around and form secondary infestations by moving it on shoes or equipment. And so the more infested areas you get, the more you will have after that. As we mentioned, these things move quickly downhill with flowing water. So if you have an infestation like we have here, you see we already have material dying down the slope. Within a couple of years, it's taken that out, and subsequently, everything from the top of that slope down to the toe of it is pretty well shot. Because you can have these surface flows, the zoospores can move with that. As we mentioned, these are very persistent in the soil, so you can't necessarily eradicate it once you get it in a large area. So your acreage becomes larger and larger that are affected, such as these infested areas here. You're not going to be able to treat it, so all you can do is try to avoid spreading it from that, and that can cramp your if you have a lot of infested material there, you're shoving it right in there with host material and something that can multiply right away. So that's potentially a much easier way to infest something than just by casually getting it in on your shoes. And we already have an increasingly wide variety of Phytophthora species going on in this native plant nursery stock. issue that we really have to get into is there's no firewall between ornamental plant nurseries and native plant nurseries. We've seen that there's sharing of material in various ways, so those pathogens can cross that border. This was a study done just recently by Jennifer Park in Oregon. They studied for a year 4 Oregon hort nurseries. They had 674 isolations, 28 different taxa in 4 nurseries. It's incredible. It's mind-boggling. And these are all introduced pathogens. So there's a wide variety of reasons why we should be concerned about this, and where possible, try to avoid getting areas infested that aren't. And so this is the question: is it really too late? And the fact is, it's really not. Compared to other situations, we still are dealing with relatively limited infestations. Places like Australia, where Phytophthora cinnamomi is a major forest disease. It's been introduced and spread along a lot of native forests. It's devastating, as they call it here, the biological bulldozer, because it affects so many of their species in these ecosystems. We're not there yet, and we don't really want to get there. And so I think this is the point of this meeting and others like it, to spread the word and not spread Phytophthora. Clive Brazier, as I mentioned, eminent plant pathologist, has written about this, and it really is a global issue. It's not just California. It really is a global problem. In terms of the nursery, the fact is it's not a new problem, and there's been kind of a lack of awareness, not the information. This 1957 BMP basically says virtually everything that's in this one. And so it basically boils down to start clean, keep it clean. There's some details, but that's really all it comes down to. And it's not technically impossible or even all that difficult, though it does cost more than doing things the cheapest possible way. In terms of wildland infestations, in sudden oak death, we have some experience about trying to stop spread. Decontamination of shoes here, that can be a kind of a team building exercise as everybody holds on to each other. Washing vehicles and keeping that infested material from going from one place to another is documented as a well-known and effective technique. And here's a paper on Phytophthora lateralis, which is it's this disease in Northern California for us. So those sorts of things can be done. Once we get infested areas, then we have to kick into yet another layer of management, and there's some of the ideas that go along here. But the bottom line, we don't want to get to the infestation if we can avoid it first, because everything becomes more complicated afterwards. So with that, I'd just like to thank some of the people and agencies that have been involved in all this research that goes into this, and that will take care of my presentation.